The overall goal of this procedure is to introduce a contusion compression clip model of cervical SCI in rats and a protocol for combined treatment involving SAP injection and NPC transplantation. First, an aneurysm clip is placed around the spinal cord and kept in place for 1 minute. This results in a bilateral contusion compression injury. Next, SAPs are injected at the injury epicenter. NPCs are injected immediately following SAP injection. An osmotic pump with a subdural catheter is inserted for the sustained delivery of growth factors. This procedure results in the delivery of NPCs to the injured spinal cord. The survival and engraftment of NPCs is supported by combinatorial treatment with SAPs and growth factors.
The implications of this technique extend toward a therapy for traumatic spinal cord injury because it provides a clinically relevant model that can be used to study cell-based combinatorial therapies that promote spinal cord regeneration.
The following experimental protocol was approved by the Animal Care Committee of the University Health Network, Toronto, Canada, and is in accordance with the policies established in the Guide to the Care and Use of Experimental Animals prepared by the Canadian Council of Animal Care. All surgical procedures must be performed under sterile conditions using sterile equipment and solutions and according to institution and government guidelines. This procedure uses Wistar rats weighing 250 to 270 grams. Before beginning the procedure,